In the previous video, we discussed all about the development of male gametophyte. So today we discuss about the female reproductive organ that is known as pistil or gynoecium. The female reproductive organ or female reproductive hole is known as pistil or gynoecium, which is otherwise known as megasporophyll. The pistil or gynoecium is the female reproductive part of the flower. Each pistil is differentiated into three parts. Each pistil is differentiated into three parts. Basal part, basal swollen part is called ovary. Middle stalk-like elongated part known as style, and the upper enlarged portion is known as stigma, and stigma is the organ of pollen receiver. A stigma is known as a pollen receiving organ. It receives pollen grain. So an enlarged basal region that is called ovary, stalk region like style, and a terminal expanded portion that is stigma. Okay. The pistil is made up of either one or more individual unit called carpel. The pistil is the whole. The pistil is the female reproductive whole, and its individual unit is known as carpel. Either pistil made up of one carpel or more than one, carpel. So when it is made up of only one carpel, it is called monocarpellary or simple pistil. When it is made up of more than one carpel, it is called multicarpellary or compound pistil. So single carpel, monocarpellary, simple pistil. More than one carpel, it is known as a multicarpellary or compound pistil. So this is all about your short note, that is pistil. Next comes to ovary. So it is very much simple. So actual part of the ovule, actual part of the pistil where reproductive activity takes place is known as ovary. It is the basal enlarged portion of the pistil. Pistil the basal part taku ovary belika kuhajaye. When we cut the ovary, we see one or more chambers called locule, or ovarian chambers are known as locule. So basal part of the pistil is known as ovary. When we cut the ovary, we see chambers. Ovarian chamber is known as locule. Inside locule, one or more ovules are present. Means locule contain one or more ovules. And the ovules are connected with ovary by means of placenta. We know ovules are connected with ovary. The region at which ovule is connected with ovary is known as placenta. And so this is just like this is the ovarian chambers. Just imagine ovarian chamber. So ovarian chamber, this is 1, this is 2, this is 3. So these three are known as locule. And inside locule, ovules are present. Here the ovule attached with ovary wall. So this wall is known as placenta. And the arrangement of ovule, you know, ovules are arranged in a definite fashion. The arrangement of ovule inside ovary is known as placentation. Arrangement of ovule inside ovary is known as placentation. So this is all about structure of ovary. Structure of ovary is a very important short note. Okay. Next comes to structure of ovule. Structure of ovule or megasporophyll. sporangium. Okay, so it is very important short note. Okay, structure of ovule. Ovule has two parts. Ovule has two parts. Basal part known as stalk and upper part is known as body. The ovule has two parts. One is a basal part or body part, another is upper part or body part. So ovule has a stalk part of the ovary is known as funicle. Ovular stalk is known as funicle. Funicle has two ends. One is basal end and another is apical end. Basal end is connected with ovary called placenta. And the apical end of the funicle connected with the body known as hilum. Okay, so this is all about stalk of the ovule. So ovule normally has two parts. One is stalk part, another is known as body part. Stalk of the ovule is known as funicle. Funicle has two ends. One end is connected with ovary. Another is connected with body. Ovary connecting region is known as placenta. Body connected end is known as hilum. So this is all about the stalk or funicle. Next comes to body. The body of ovule has two ends. One is chalazal end. Another is micropylar end. Body of ovule has two ends. One is chalazal end. Another is micropylar end. It means body of ovule becomes polar. Next, body is differentiated into two parts. Body is differentiated into two parts. One is body covering. Another is body proper. Body covering of the ovule is known as integument. Body covering is known as integument. Integument either one layer or two layer. Integument either one layer or two layer. When it is one layer, it is called unitegmic. When it is two layer, it is called bitegmic. Okay. Unitegmic or bitegmic. Unitegmic is the characteristic feature of gymnosperm, whereas bitegmic integument is a characteristic feature of angiosperm. Okay. So a short question है प्राय टाइम ऐसे unitegmic and bitegmic ovule is the characteristic feature of gymnosperm and angiosperm respectively. Okay. The continuous end of the integument is called chalaza and the discontinuous end of integument is called micropyle by leaving a small pore. Okay. तो ओव्यूल को पोलारिटी दबा काम किए करे इंटेगुमेंट ही करिताए एंड इन फ्यूचर इंटेगुमेंट प्रोवाइड सीड कोट. Integument provide seed coat. Okay. Next, inside integument a large mass of parenchymatous cell present. That parenchymatous cell is known as nucellus, and the center of the nucellus contains a sac. This sac is known as embryo sac, and inside embryo sac egg present. एम्ब्रियो सैक भितर एग तुम देखुच तो बाहर भीतर साइड को जिते जीव फास्ट देख इंटेगुमेंट इंटेगुमेंट पर आसब न्यूसेलस न्यूसेलस पर आसब एम्ब्रियो सैक एंड एम्ब्रियो सैक भितर देख तुम कौन देख एग को देख दैट इज द फीमेल गैमेट तो एम्ब्रियो सैक डेवलपमेंट को फीमेल गैमेटोफाइट डेवलपमेंट बेलिका कहुए एम्ब्रियो सैक को ही फीमेल गैमेटोफाइट बेलिका कहुए तो आई ऑलरेडी टोल्ड दिस अबाउट दिस नोट ऑलरेडी कही सारी नोट जहाँ विषय अच्छी विषय ऑलरेडी कह सारा सबूक जिनस कहुई ओके ओके न्यूसेलस देन दिस इज द टाइप्स ऑफ ओव्यूल टाइप्स ऑफ ovule. There are normally 6 types of ovules are seen in nature. So on the basis of arrangement of micropyle, chalaza and funicle. On this basis, ovules are categorized into 6 types. So number 1 is orthotropous type, which is known as erect ovule. This indicate micropyle, chalaza and funicle present in the straight line, and this erect ovule or orthotropous type is very commonly seen in case of gymnosperm. Next, Anatropus ovule or inverted ovule. This type of ovule is the inversion of orthotropus into 180 डिग्री तो जिते ओव्यूलटा इरेक्ट ओव्यूल इनवर्सन हम 180 डिग्री को ताले सेटा कौन हम इरेक्ट इज इन्वर्टेड तो ऑर्थोट्रोपस इज कन्वर्टेड टू एनाट्रोपस तो ऑर्थोट्रोपस हमें देखी कैरेक्टरिस्टिक फीचर ऑफ जिम्नोस्पर्म एनाट्रोपस ओव्यूल व्हिच इज द कॉमन टाइप ऑफ ओव्यूल जहाँ विषय हम वर्तमान पढ़ुले भी तो सेटा होची कौन इन्वर्टेड ओव्यूल कैरेक्टरिस्टिक फीचर ऑफ एंजियोस्पर्म एटा कौन इंडिकेट करुच्ची माइक्रोपाइल चालाजा गोटे स्ट्रेट लाइन रे रहउचंती माइक्रोपाइल गोटे साइड रे रहउची फ्यूनिकल नेक्स्ट कम्स टू हेमिट्रोपस ओव्यूल हेमिट्रोपस इंडिकेट H for hemi, H for half inverted ovule means micropyle chalaza funicle sangare 90 degree angle formation karibe so it is half inverted hemitropus ovule. Next comes to Campilotropus. Campilotropus ovule indicate the body or the nucellus. Here the body or nucellus is carved into horseshoe shape तो घोड़ार नाल घोड़ा तले जो देखि पाद तले गोटे प्लेट लगी थाए ताको हॉर्स शेप बेलिका कह जाए इवन इफ तुम मैग्नेटिज्म जिते पढ़ी हॉर्स शेप गोटे मैग्नेट विषय भी पढ़ी तो यहाँ तुम हूँ कैम्पिलोट्रोपस ओव्यूल बॉडी बा न्यूसेलस कर्व हो हॉर्स शेप सो दिस इज ऑल अबाउट द कैम्पिलोट्रोपस. देन कम्स टू एम्फीट्रोपस. एम्फी माने होची बोथ तो हियर both body and embryo sac here body along with embryo sac becomes carved into horseshoe shape so it is called amphitropus. And the last type of ovule is known as Campilotropus. Here the funicle is much more elongated so it rotate or it curved around a full circle around the ovule. तो जिते बेले ओव्यूल चारिपटे फ्यूनिकलटा 360 डिग्री कवर करे सेते बेले ताको कहा जाए बा फुल सर्कल कवर करे सेते बेले ताको कहा जाए Campilotropus. तो फ्यूनिकल कॉइल द ओव्यूल कॉइल अराउंड द ओव्यूल जेहेतु गोटे फुल सर्कल कवर करे सेति पे ता ना हला Campilotropus. So this is all about your short notes, various short notes. So next comes to second long question, that is the development of female gametophyte. Development of female gametophyte. Next, It's a very simple long question, just like microgametogenesis. तो फीमेल गैमेटोफाइट डेवलपमेंट मेल गैमेटोफाइट डेवलपमेंट सांगे पाखापाखि सामान आसब खाली कंपैरिजन करी पढ़िले हैला। तो देख लिखला द मेगास्पोर इज द फास्ट सेल ऑफ फीमेल गैमेटोफाइट और एम्ब्रियो सैक। तो आम देखुचे मेगास्पोर नथिले मेगास्पोर रु आम एम्ब्रियो सैक पाउचे। तो मेगास्पोर नथिले एम्ब्रियो सैक मिलबनी कारण आम ऑलरेडी जानिचे मेगास्पोर रु एम्ब्रियो सैक रो फॉर्मेशन हबो आ एम्ब्रियो सैक रो आ गुटे ना होची फीमेल गैमेटोफाइट। ओके तो यही प्रोसेसटा दुईटा स्टेप रे कंप्लीट होची, गोटे होची मेगास्पोरोजेनेसिस गोटे होची मेगा गैमेटोजेनेसिस। तो फर्स्ट स्टेप कहला द फॉर्मेशन ऑफ एम्ब्रियो सैक और फीमेल गैमेटोफाइट फ्रॉम मेगास्पोर इज नोन एज female gametophyte, and this process is completed within two important steps. One is megasporogenesis, another is megagametogenesis. This all total process occur inside the nucellus. So first step, megasporogenesis. So megasporogenesis is the process where megaspores are produced from megaspore mother cell. Megaspores are produced from megaspore mother cell. It is called megasporogenesis. What happened? Initial state. At initial state, some of the hypodermal cell of nucellus, hypodermal cells of nucellus act as archisporial initial. Archisporial initial. This archisporial initial differentiate from other cells by means of its dense cytoplasm and prominent or large size nucleus. Archisporial initial separated itself from other cell by means of dense cytoplasm and prominent or large size nucleus. The archisporial initial give rise to megaspore mother cell. Archisporial initial in future रे कौन दबा megaspore mother cell को convert हो जिवो. Archisporial initial then converted to megaspore mother cell. The archisporial initial, though archisporial initial diploid, megaspore mother cell is also diploid. Then megaspore mother cell undergoes reductional division or meiosis to produce 4 megaspores arranged in linear order. Arranged in linear order. तो जेहेतु चार्टा प्रोडक्शन होचे, so it is called linear tetrad. तो archisporial initial निजे एक्ट कला मेगास्पोर मदर सेल. देन मेगास्पोर मदर सेल कौन कला मेयोसिस कला प्रोडक्शन कला 4टा मेगास्पोर अकर इन लिनियर ऑर्डर. तो 4टा माइक्रोस्पोर वर्तमान मेगास्पोर वर्तमान टेट्राएड रे प्रोडक्शन होइचंती. नेक्स्ट आसिला ऑफ व्हिच तो 4टा रो हमर गोटे रहिचंती माइक्रोपाइलर एंड आडो को चला जा एंड आडो को रहिचंती. दीटा एंड तो ओव्यूल पोलारिटी अछि माइक्रोपाइलर चला जा, ऑफ व्हिच the chalazal megaspore 1 becomes functional and the rest 3 becomes degenerated, and these functional megaspore, chalazal functional megaspore, help in the production of embryo sac. Okay, so next the megasporogenesis complete. Next comes to megagametogenesis, the process by means of which megaspores are converted to complete embryo sac which contain egg is known as megagametogenesis, or in the other sense we told the formation of egg inside embryo sac is known as megagametogenesis. We know common type of embryo sac that are found in nature is known as polygonum type. Common type of embryo sac development seen in nature is known as polygonum type. Here, this polygonum type nucleus, polygonum type of embryo sac bear 8 nucleus. Polygonum type of embryo sac bear 8 nucleus, and these 8 nucleus are distributed 4 at each pole. 4 at each pole. For the production of 8 nucleus, a megaspore requires 3 mitotic division. Megaspore by embryo sac ru megaspore ru 8 nucleated embryo sac conversion हबा पाई गोटे मेगास्पोर को 3टा माइटोटिक डिवीजन को फॉलो करिया पड़िबो, जहां पर से 8 न्यूक्लिएटेड एम्ब्रियो सैक जो थरे कि 4 at each pole रहबे, प्रत्येक गोटे रहबे 4टा रहबे माइक्रोपाइलर, 4टा रहबे चालाजाल. तो एभली टाइप रो एम्ब्रियो सैक को पॉलीगोनम टाइप लिखा कहा जाय. mitotic divisions are not followed by wall formation. जो तीनटा successive mitotic division हबो एतर कौन से wall रा formation हबो नहीं बा cell wall रा development हबो नहीं केवल multi nucleate condition ही देखिबा को मिलिबे. तो देखो फिगर देखले बुझ परिबो. तो मेगास्पोर फंक्शनल मेगास्पोर फास्ट माइटोसिस, मेगास्पोर सेकेंड माइटोसिस रे प्रोडक्शन कला 4टा न्यूक्लियाई एंड थर्ड माइटोसिस रे प्रोडक्शन कला 8 न्यूक्लियाई 4 at each pole. Okay, there is no wall formation occur in between the nucleus. Then what happened? Out of the 4 nuclei from each pole, one comes to the center. तो प्रति पोल रू जो 4 न्यूक्ली थिले प्रति पोल रू गोटे न्यूक्ली वर्तमान सेंटर को चली आसिले एंड एक्ट कले पोलार न्यूक्ली. तो सेंटर कंटेन 2 पोलार न्यूक्ली एंड द पोल कंटेन 3 each. प्रत्येक रू 3 रहिले. Now after this distribution cell wall formation or cell wall laid down by cell wall formation begins. So it produces 3 cell at micropylar end, 3 cell at chalazal end and 1 cell at the center. So nuclear जदि distribution पचारे the nuclear distribution embryo sac भीतर रहबा 3 2 3 and the cellular distribution in embryo sac 3 1 3. Okay, next the 3 micropylar cells are known as egg apparatus, of which the central large one is called egg and the 2 lateral one is called synergid, and the chalazal 3 are known as your antipodals and the central 2 polar nuclei surrounded by a single cell that is known as central cell. So a mature embryo sac contain 8 nucleus and 7 cell. So mature embryo sac that is polygonum type contain 8 nucleus. Right time question आसे बोल सुनो 8 nucleus 7 cell. Very good. तो सॉरी गला तो हमारा वर्तमान कंप्लीट है गला. लॉन्ग क्वेश्चन भी कंप्लीट है गला. सेकेंड लॉन्ग क्वेश्चन कंप्लीटेड. ओके, डेवलपमेंट ऑफ फीमेल गैमेटोफाइट. ओके, नेक्स्ट और लास्ट टॉपिक दैट इज structure of embryo sac. It is a very important short note. Very important short note. In angiosperm or the female gametophyte is the embryo sac. Female gametophyte is otherwise called embryo sac. Embryo sac is oval. Embryo sac is normally oval in structure and it contains multinucleated multicellular haploid structure. A mature embryo sac contains multicellular because it contains 7 cell. Though it contains 7 cell, obviously it is multicellular haploid structure. Though it produced through meiosis, so its cells are haploid. Okay, next, embryo sac membrane is derived from megaspore. We know megaspore is converted to functional megaspore is converted to embryo sac. So embryo sac membrane comes from megaspore membrane. Okay, the common type of embryo sac is angiosperm is a polygonum type which is approximately 80% seen in 80% flowering member. It is 8 nucleated 7 cell structure of which 7 cells are arranged 3 micropylar, 3 chalazal and 1 central. Then 3 micropylar 1 act as egg apparatus of which middle one is large and that large cell is known as egg or oospore. Okay, and the 2 lateral one is known as synergid and the synergid has a special appendages and these appendages is known as filiform apparatus. Filiform apparatus which is a characteristic feature of your synergid. Next, 3 chalazal cell, the of which 3 chalazal cell of the embryo sac is called antipodals and these antipodals degenerate just after fertilization. Next, last one, the central cell which is the largest cell of the embryo sac which contain 2 polar nuclei and by the by using two polar nuclei, a single central cell is produced at the center of the embryo sac. Okay, this is all about our topic.